Living things produce a very nasty chemical called hydrogen peroxide. To break down hydrogen peroxide, the living things have catalase. So catalase is the enzyme we're going to use. We'll use some celery, and to get us some enzyme, we'll chop up the celery. And transfer it to a blender. So we add in some water. And we blend it for 4 seconds. To remove all the cell debris and the cell parts, etc., we pass it through coffee filter paper. And that will give us our enzyme here. The filtrate will be our enzyme. We transfer the filtrate containing the enzyme to a beaker. And this time we want to take half of this and boil it. So we take half of this solution into a beaker and we'll boil it. Having allowed it to boil for a number of minutes, we remove it from the hot plate and we place it there. And we take our unboiled solution. On this side we have the unboiled enzyme, and on this side we have the boiled enzyme. We take 10 cubic centimeters of the unboiled enzyme and transfer it to the graduated cylinder. We take 10 cubic centimeters of pH buffer 10, which is the optimum pH for catalase, and transfer it in. And we take our 2 drops of washing up liquid. On the other side, we take 10 cubic centimeters of the boiled enzyme. We take 10 cubic centimeters of the pH buffer 10. And again, 2 drops of washing up liquid. Again, hydrogen peroxide is dangerous, so wearing gloves and goggles for safety. And transfer the hydrogen peroxide into a beaker first. And we pour 5 cubic centimeters of hydrogen peroxide into the small graduated cylinder. And also 5 cubic centimeters into the second graduated cylinder. Now, we want to run everything at the same temperature. Catalase works best at 25 degrees centigrade. So we transfer both of these into the water bath at 25 degrees centigrade. And both of these into the water bath at 25 degrees centigrade. And we leave them there for 10 minutes to come to the same temperature. So everything will be at the same temperature. After 10 minutes in the water bath, we remove the boiled enzyme and the hydrogen peroxide. And we mix the peroxide, being the substrate, into the boiled enzyme. And no foam is produced. Because of the high temperature, the enzyme is denatured, it has lost its shape, also lost its function, doesn't work. No bubbles, no foam produced. If we take out the unboiled enzyme, and again mix the peroxide, straight away foam is produced. Oxygen bubbles are produced down here. They interact with the washing up liquid and foam is produced. We wait 2 minutes and calculate the volume of foam. Again, subtracting the volume of liquid in the bottom from the foam at the top. So 2 minutes later, we'll get our results.